film a couple things today, so it's not a different day. I am wearing the same clothes on the same day. So let's talk about how is this controlled? How does this happen so perfectly all the time? And what happens when it doesn't? So when the cell cycle to make body cells, those that help us grow and heal, doesn't go right, that's when tumors can form. And this is where it becomes so fascinating to think about all of this. So let's start talking about what controls this cycle. And it's really important to remember it's just that. It is a cycle. It is continually happening in your body. Not every cell goes through the cycle at the same speed, which we'll talk about in a few minutes. Some are really slow, some are really fast, like our skin. But one thing that happens is it's very important. Another reason a balanced diet is important. We have certain molecules in our body that help this happen, and the main group of molecules are proteins. So we're going to talk about two proteins that make this cycle start, and then we're going to talk about one protein that makes it stop. And then we're going to talk about a couple other things that our cells count, if you could imagine packing for an amazing trip, making sure you have all the right stuff in your suitcase because you don't want to get to your destination and forget clean underwear. I mean, seriously, right? So, the first things we're going to talk about are checkpoints in the cell. So this is a different picture of the cell cycle, and it includes the checkpoints. So there is the G1 checkpoint, and this is really cool. It looks for mistakes. It looks to see if there are enough growth factors, which we'll talk about in a few minutes. It's just kind of like that's food for the cell to have energy to get through this process. And then other nutrients that are needed. This is a resting phase, which we'll talk about in a minute. It's really not rest. It's just some cells, their cell cycle, this whole process is so slow that they sit here for a little while. And we'll talk in a minute about that. If they get past the G1 checkpoint, then they go to the S phase and they make more DNA. They go to G2, and right before M, or mitosis, where the cell goes through prophase, metaphase, anaphase, and telophase, they go through the G2 checkpoint. And this is really all this cell is doing is saying, do I have enough of everything to move forward? Is my suitcase fully packed? And then at one point, which we will talk about, during metaphase, it's going to count one more thing. Because if it doesn't have it, it knows that all the chromosomes are not at the equator, and they are not ready to travel into the two brand new, beautiful, identical cells. And that's what we want when we get done with this. So, the checkpoints, the G1 checkpoint, this is the restriction point. And that's why it's called, on some of the things you will see, an R for resting. Really, it's restricting itself from moving forward because it doesn't need to yet. It doesn't need to yet. Oops, I went backwards. So if it's given, it's ready to go forward to the S phase and move on. There's called a G0 phase, or that resting phase. Really, it's a restriction phase. Resting, I think, is a little outdated. The cell's not at rest because it's still doing its job. And two cells to think about here are liver cells, which their life cycle is about a year if they're damaged. Once your liver is fully formed, if you don't damage it, they don't have to fix themselves. But unfortunately, we do sometimes. If we take Advil or Tylenol, other medications, right? If we eat a really high sugary diet, our liver has to work harder, so we have to replace those cells. Drinking too much alcohol can cause this to happen too, and that can permanently damage it. But they go into this non-dividing. They don't need to divide. They can do their job without it. They're tough, just like our nerve cells. Right, their life cycles are about 50 years to your lifetime. So that's why if you cut your spinal cord, it can't heal because its life cycle is so long. It takes it forever to get through this phase. And I like to tell a story about my dad cutting his hand really bad when I was little, and he hadn't had feeling here for a really long time. We were trying to bust open a coconut, and he sliced it open, had to get a bunch of stitches. And now, all these years later, he has tingling starting again because those nerves that he severed deep in his hand are now starting to heal and rejoin, and he's getting these weird phantom, they're really not phantom, I guess, real feeling back in that spot that had been numb for all those years. So, how does a cell know to do this? How does it know to go through this cycle? Right? How does it know? So 2 things that it needs to do this. Don't forget to watch this. It needs cyclins and cyclin-dependent kinases. So has 2 things. Both of these are just proteins. The first one, protein kinases, are enzymes. They're going to activate. Remember, enzymes speed up chemical reactions. In biology, they control them. And in this case, they're going to make them go or they're going to make them stop. So these are enzymes that if they're phosphorylated, they're going to be able to go ahead. So if that enzyme is phosphorylated, which means a phosphate group attaches to it in a chemical reaction during this process, it's going to make it go forward. We talked a lot this year about things getting phosphorylated. ADP becoming ATP. So certain kinases allow for G1 and G2, so both growth phases to start. We are looking for those specific kinases, those specific proteins, and if they're not there, it's not going to go forward in the cycle. So it has to have these kinases. But to make them happen, a cyclin has to attach to them. I know I'm doing it by myself. Are you proud? So this is just a protein that's named because it cyclically fluctuates in your cells in concentration, and when it's there in a high enough concentration, the cell can go through its life cycle, and if it's not, it can't. Don't worry, it's always floated. Those proteins are always available, right? It just has to attach, and the minute it attaches, it will move forward. So both of these are necessary to go from G1 all the way through to G2. And these are some of those things, right, those kinases and the protein, or cyclin-dependent kinases and the protein kinases, these are here in those nutrients and growth factors that we're looking for. Those allow this to go forward. So those 2 things allow the cell cycle to go forward in a healthy cell. Here are 2 graphs I'm not going to go over right now to keep this video as short as possible. You can analyze these if you want. This one showed up on an AP test years ago. I doubt you would ever see it on the 2 FRQs that are coming up. So if we have protein kinases and CDKs, it can go, but how does it know to stop? And this is super important. If it can't stop, a tumor will form. And to get it to stop, we need proteolytic enzymes. They literally cut the cyclins. They break down the cyclins that are around the cell, allowing it to divide. So these are enzymes that destroy these 2 things. So they cut them up, and the cell stops dividing, and it goes on doing its cell job. Maybe it's a heart cell. Maybe it's an eye cell. Or maybe it's a skin cell. Maybe for someone fighting this awful virus that we're all stuck home for, it's their immune cells, right? They're having to reproduce white blood cells in order to attack this horrible virus that's slowing us all down and keeping us all home. So if there are proteolytic enzymes, they're going to cut it. The cell will go through mitosis, and it will go right back into interphase and continue its cell life cycle, making healthy new cells. So there are a couple other types of signals that happen that allow cells to grow properly and to multiply properly. And some of them are inside the cells and some are outside the cells. I like to think of them like traffic lights. You can go, you need to stop, you can go, you need to stop. So the only internal signal is what I was talking about during metaphase. The cell comes these things called kinetochores. So when the chromosomes head to the equator, to the middle of the cell, they form these little structures called kinetochores where they stick to the spindle fibers. I'm going to skip way ahead to this picture. So this is where the kinetochores would be, stuck to here. If this reaction, this is just a little chemical reaction that takes place between the chromosomes and the spindle fiber, these little brown lines, it counts, and if it doesn't have enough of those, it's going to stop. It's like, whoa, right, suitcase is missing something, I'm missing something. So it's going to stop, And in humans, we would want 23 of these kinetochores. No, 46 kinetochores. Sorry, I was working on meiosis this morning, which we'll get to way later in the week. So we're going to want 46 of these. We need all of these pairs to make sure that we have enough in each body cell. So we're going to have 46 pairs of chromosomes because we need 46 chromosomes in each new cell. So it's going to count all of those up. Right? In some insects, like the fruit fly, there's only 4. Right? They don't have a lot of chromosomes. But we have a lot of chromosomes. Alright, going back. So that's the internal signal. It's counting up to make sure there are enough pairs to move forward with division. But this is where it gets really cool. There's external signals around the cell that either tell a cell, I have to divide because I'm not touching another cell, there's something wrong, or I'm touching too many cells, I'm feeling crowded, stop. So one of those is growth factors. This is released by some of your body cells that tells it to divide, so it's time to grow. Then cells are density dependent. Cells hate being crowded. Our cells just want to touch certain number of cells. They don't want to have pressure on them, so they stop dividing at that point. They don't like to be crowded. They have serious space issues. That's how I ended up with this, because I don't like crowds, but I still took my daughter to Universal Studios, so I bought myself this shirt. Alright, so they're density dependent, and they're also, though, anchorage dependent. Cells like to touch other cells, unless they're blood cells. Blood cells are in a liquid, it doesn't matter. But all your other body cells like to touch cells. So the minute you get a cut, that tells those cells, holy moly, I'm not touching any other cell. Right? I want to make new cells to seal that cut up. So they're anchorage dependent. They want to be attached to something. They don't want to be crowded, though. They want to be attached. Cancer cells, on the other hand, don't exhibit density dependent or anchorage dependent. They can go off as loners, or they can crowd up. Normally, remember, our immune system will catch those cells that there's mistakes in and it will destroy them. But if it doesn't, a tumor can form. And there's 2 types of tumors. Benign, they're an abnormal clump, but it won't spread, or malignant. And if you want to think about malignant, you can go all the way back to the very first slide of all of these slides. And if we had class together, we would have discussed this, but this is a malignant tumor forming. So here you have normal cells, a cell that goes wrong, that cell multiplies and makes this clump, and to make it malignant, then this breaks through this membrane and it enters a blood vessel and it can spread in your body. So, just like I did yesterday, where I posted the mitosis review slides, and I put a live document in there that you can ask questions on or comment so we can have a class conversation. I'm going to do the same with these. I'm going to wait a couple days and let you guys work through all of this. Make sure you're double checking all of your vocabulary against what I've talked about that I assigned. Make sure you ask me questions, and make sure that you are commenting in the AP classroom that you have watched these, because that is how I'm checking in your work right now, just to make sure that you're keeping up. And remember, this is about being college ready. I will only be posting one more lesson this week, because I know this is a lot, especially if you just started this week. Kids that previously started already have the vocab and things done. But we're gradually working through this. So we'll talk to you guys soon. Bye.